In the previous part of our lecture, we have discussed about the basic principles of Mendel. Now let's look at extensions to Mendel's principles because Mendel's work mostly work on centered rather on plants. And many geneticists, many scientists after his generation have tried replicating his experiments on different organisms, especially for animals, with varied successes. And they have seen that there are more to Mendel provided the groundwork, but there are more to it than meets the eye. So it's not as simple as those three. So the first example here is this puppies, this litter of Labrador puppies. So according to Mendel, we mostly have this traits, two copies of the gene. But in here, we found out that we have three colors of of a litter of puppies. These siblings containing three different coats of color. So what is actually going on here? According to the dominance, it should either be black or white, or basically the golden one. But we have a brown. What does that mean? So in this part of the lecture, we will look at some extensions to Mendel's principles. So first half here, we have here is the dominance relationship. So it is not always, or there are cases that um, the dominance is not exactly complete. So for example, you have here a trait, let's say a white a color, whatever color that is. So a white color and a blue color. So a complete dominance will show that if you cross a pure white, a genotypic homozygous white with a homozygous blue, whatever is the dominant trait will appear on all of the offspring. So basically here, if the dominant one is the white, you will get a white colored offspring. If the dominant one is the blue, you get all of offspring to be colored blue. However, in cases of what you call incomplete dominance, just like those Labrador puppies we shown earlier, we have here a cross between a homozygous white and a homozygous blue, but the color is a mixture of. So it's called an incomplete dominance. There's a mixing. Another one is a codominant scenario. In the codominant scenario, again, there's two colors, but instead of basically just mixing them with basically like a colored paint, you mix blue with white, you get up with the light blue. This one has both blue and white colorations on them. So this is a codominant relationship. So another example for this types of dominant relationship. For the incomplete dominance, in here, the description is, by the direct definition of incomplete dominance, is that the offspring, the F1 hybrid of these two homozygous traits, homozygous for these traits, resemble neither of the pure breeding parent. So for example, a red flower and a white flower provides you with a pink colored offspring. So it's neither white nor is it colored red. So it's colored pink. So basically, this is incomplete dominance. So both of them are dominant, but it's an incomplete dominance. So how is it different with this codominance? So for example, an incomplete dominance, if we relate them to biochemical terms, for instance, or rather, since uh, it really relates back to biochemistry. So for example, here in this colored plants, you have an enzyme that produces the red pigment and an enzyme that produces the white pigment. In an incomplete dominance, you have, or rather, sorry, an enzyme that can generates the red pigment, and then the other one is no pigment at all. So having just one copy of the gene for this red pigment gives you a copy of, or rather a color, a phenotypic expression of a pink one, because it only has one copy, and that is not enough to actually produce that vibrant red color. That's why it's colored pink. So that's one way, the biochemical description or the biochemical basis of that. So in order to have this vivid red color, you must have two copies, but you only have one copy, so it's an incomplete dominance. You basically just have a watered down color of red, or rather a more lighter version of red, which is pink. Now we have this another one which is different, entirely different, is codominance. Codominant is different from incomplete dominance, mind you. So in the codominance, the offspring, the F1 hybrids, display traits of both parents. The best example for this one is blood types. So we have two dominant blood types, the A and the B. Now, if you have this uh, an A, or rather a gene for the A blood type, that means you have an enzyme or set of enzymes that produces, because the blood type here are the glucose, the oligosaccharides at the surface of red blood cells. And the trait or the gene encodes for the enzyme that creates this specific pattern of oligosaccharide. So blood type B is also another different set of patterns. So A pattern, B pattern. And so if you have a blood type A, you basically have only A pattern. Blood type B, you have the B pattern. But for blood type AB, which is the offspring of an A and a B, the dihybrid, the hybrid of them, it contains both A and the B. So it has this enzyme for the both patterns. And the AB blood type contains both. So the surface of their blood cells have both patterns on the surface of the red blood cells, and that is codominance. Both of them occupy on the same position. So basically, they have both patterns on the surface. So that's codominant. In contrast to incomplete dominance is that for this vivid color to appear, you must have two copies. But since you only have one copy, you get a lighter version. So its expression is it does not resemble technically the parent. This one resembles both parents. It has the blue and it has the red patterns of this blood type. Now, another concept here is polymorphism. So you have polymorphic genes. In the case of Mendel, he is actually quite lucky to choose the traits that some critics actually criticize Mendel having this because the results is very ideal. He got lucky for choosing the traits of the two alleles. But we have traits that have more than two alternatives, and we usually see them in animals. So we have more than two alternatives that exist and are common in the population. So an example for this one is the ABO blood type. We have the allele for A, the allele for B, and the recessive allele for O. Basically, none of the enzyme for the surface of the blood cell. So we have three alleles here. Now, another one here is for the rabbits. So you have the black, the chinchilla, Himalayan, and the albino colors. So you have four different traits. Now, because you have multiple, it's not only just two, but multiple types of alleles, we can have this dominance series. It brings to the concept dominance series. So we have dominant series, which is the order of your dominant alleles. Which allele is more dominant than the other? And in order to do that is, of course, you need to breed them. So, for example, we have an agouti, the common color of a mice, which is agouti. Another one is a mouse with a black mouse with a yellow belly. So crossing them produces an agouti. So that means agouti is dominant to the black mice with yellow belly. Crossing agouti with a purely black mice also produces an agouti. So the agouti is also dominant over the black. Now, crossing the black mice with a yellow belly and a completely black, basically a pure black mice, produces a black mice with a yellow belly. That means the black mice with a yellow belly is dominant over the completely black. So you have a series of dominants here, whereas the agouti color is the most dominant, followed by the black with a yellow belly, and lastly by a completely black mice. So you have this order of dominance for these traits. Now, aside from this one, we have wild types and mutants. So you will encounter common terms like wild types, mutants, mutations, etc. So when we say wild type, it's the allele that occurs most frequently in the population. We usually consider it normal, and this wild type allele is usually the dominant allele. So and because it's dominant because it's usually seen in the population. So it's usually arbitrarily designated as a normal one. And mutations are changes to genetic material. So sometimes we have this random mutations that occur. So for example, here is the uh the mutation here is a melanoma. You have a jaguar and what you call a black panther. In reality, a black panther is not actually a separate species, but rather it's a type of feline. It can be a tiger, it can be a jaguar, it can be what else? A lynx, a feline variety that has a mutation. And because of that mutation, it produces a very dark pigments. Hence, you have this black cat, a black large cat. So this is also another jaguar. So by species, this is a jaguar, but it's colored black. So because of mutations. So mutations are not necessarily bad because they provide us with changes, variation, a source of new variabilities or variations in the population. Although they occur in less in frequency, if that mutation becomes dominant, sometimes it becomes the wild type eventually because it takes over the population. So some mutations are good, some mutations are bad. That gives us in this several types of mutations. So we have loss of function mutation, which results in diminished or reduced activity. So loss of function is actually bad, mostly bad type of mutation. Then we have gain of function mutation, which results in enhanced or additional functions or property or characteristics. And neutral mutation, which is neither good nor bad. So it's neither a loss nor a gain. So an example of mutation here is the mutation in the hemoglobin that produces the sickle cell. So it can be considered a loss of function mutation because um, of course, if you have sickle cells or you get an anemia, it's called the sickle cell anemia. So mutations on the hemoglobin makes your blood inefficiently carry oxygen. So it cannot efficiently carry oxygen. So you end up with anemia. Whereas however, a gain of another side for this change can also be considered a gain of function because it found out that people with sickle cells are less likely to die of malaria. So it's bad because you have anemia, but you have resistance to malaria. So that is another way to look at that. So gain of function provides you with a certain immunity or resistance to a certain something. So those are some types of mutation. And how can mutations happen? So here are some mechanisms of mutation. We usually designate them as chromosomal mutation, the one on the left, and the DNA mutation, the one on the right. So let's look first at the DNA mutation. So if you remember the structure of the DNA, so you have a sequence of your oligonucleotides. So we have three basic types. You have substitution. So one nucleotide is substituted over the other. You have G to C here, mutation. So there's a change in the sequence or rather change in the letter. And insertion is you insert a nucleotide. So there's a movement. Deletion is you delete a nucleotide, just like this one. T is deleted. So another movement here, insertions and deletions. So insertion and deletion mutation are usually very impactful because it changes the codons downstream. So unlike substitution, it only affects that localized region. So, or at least generally for that sequence, although depending on the substitution, it can impact the overall structure of the polypeptide. But that's another discussion for a later date. Now, these are DNA mutations. You can see here it's called DNA mutations because it impacts directly the DNA. Now, chromosomal mutations on the other hand have a more greater scope. In here, you have the actual whole chromosomes and deletion actually similar. So you have a deleted letter B. So one part is deleted. You have replication. One segment is replicated or copied twice over. So you have the replication of a segment. You can also have inversions. So you have there's an inversion of this side of this. If this is the series, you have an inversion, the genes it becomes inverted. There can also be translocation. Two different chromosomes translocated over the other. So you have a large translocation here. In fact, chronic CML, chronic myelogenous leukemia, is caused by a translocation of the chromosome. So that's cancer. Okay, that gives us to the topic of chromosomes and genes because we're talking about mutations. So, previously we have been talking about Mendelian principles. This becomes the theory of inheritance known as the chromosomal theory of inheritance because we are looking here now at chromosomes and the genes. In Mendel's time, the DNA is not yet discovered. They still do not know. It's the trait is just described as a factor that's being inherited from parent to offspring. But right now, because of our discoveries of the genome, we are the recent discoveries in molecular biology, we know the physical manifestation or the physical entity of this unit factor. And these are the genes, which are encoded in the chromosomes. So in here, actually images of the fish. Fluorescence in situ hybridization. So these are your chromosomes, and the bright red lights here are basically uh, basic genes, or rather, these are the genes or um, regions, hybridized region of the DNA. So it shows genes found in your chromosomes. So chromosomes, basically in the chromosomal theory of inheritance, chromosomes are the carriers of genes, and that meiosis is the physical basis of Mendel's postulates. So what is a chromosome? Chromosome, we usually see them in the microscope during mitosis or meiosis. This is your chromosome. So you form, if we look closely at this chromosome, it forms your chromatin structure, which is basically a DNA and histone proteins. Your DNA is basically a very, very long thread-like molecule. It's like a thread. It's wound, it wounds around a histone that's arranged properly to form the chromosome. But the chromosome is basically just like a spool of thread, and that thread is your DNA. And our genes are actually mapped to sequences or segments of that long, long, long string of DNA. Genes are segments of that encodes for specific trait, and we can actually map them. So this is as an example of a genetic map. So this is your chromosome. You have those marks. Those are the genes that marks where your basically the genes where it is found in your chromosome. So further experiments on fruit fly also. Usually, it started by Thomas Hunt Morgan, and it gained a large following. Many people have replicated actually not only on fruit flies but also in zebrafishes, Arabidopsis plants, on more to better understand how genes are um how genes are being transmitted across the generations, and at the same time how genes are being expressed on that particular individual. So in Mendel's experiment of a fruit fly, it follows a concept of Mendel, so such that in the hybrid crosses you have this recessive types that is smaller in percentage. However, because animals, the males and the females are very much uh, are overtly observed. You know which is male, which is female immediately. So there, and coming with the chromosomal theory of inheritance, there now appears a concept of linkages. So aside from the law of independent assortment that the genes are assorted independently of one another, but the way the genes are expressed, there are some genes that are linked to the X chromosomes, the differences in the gender. So some genes are expressed because of some differences in the gender. And at the same time, some genes are transmitted through gender differences. And not only that, some genetic linkages, because genes are physically present on your chromosomes. How the chromosomes are transmitted actually puts into question independent assortment. So some genes, because they appear on the same chromosome, they can be transmitted simultaneously. They are always together, or usually tend to always be together when they are being transmitted instead of being assorted independently, although they are independent of one another. So these are further studies. Basically, the chromosomal theory of inheritance went beyond Mendelian's ideas and more into the physical mechanisms on how our traits are transmitted from parent to offspring and how these traits are actually expressed. You have this gene, but are you expressing that gene? What are the mechanisms that expresses your genes? And those are quite heavy topics, and these topics we will be discussing on the next lecture. But for now, these are the two types we have discussed, the two principles or two theories for your the two theories of inheritance, the Mendelian and the Mendel's theory, and of course the chromosomal theory of inheritance. So more on chromosomal theory of inheritance on the next lectures.